introduction, and first I'd like to thank the organizers very much for the invite and the organization of this meeting. It's really an amazing meeting, both scientifically and socially. I will speak on antibiotics, and the reason why I'm going to speak on antibiotics is the antimicrobial resistance crisis. You're all familiar with this. So, according to a recent report commissioned by the British government, antimicrobial resistance will become the killer number one in the near future, causing more annual deaths than cancer. And already right now, the annual mortality of antimicrobial resistance is larger than the combined annual mortality of malaria plus HIV. So this is huge, this is urgent, and if we want to fend off antimicrobial resistance, well, then we need new antibiotics that use new mechanisms. So in my group, we work on a small number of antibiotics, and what all these antibiotics have in common is that they specifically target lipids that only exist in bacterial membranes, but not in human membranes. And these antibiotics are powerful and they kill a broad spectrum of superbugs. And since their lipid targets are conserved and much more difficult to modify than proteins, resistance development against these antibiotics is generally slow. But there is one catch. Currently, we cannot efficiently develop these compounds towards clinically useful antibiotics because we don't understand their molecular mechanisms. And this molecular void, this lack of molecular knowledge, is the real bottleneck for medicinal chemistry. So we would love to change this and would love to understand these powerful compounds, and therefore, to this end, we have designed an integral structural biology approach, and I'd like to show you how this works on the example of drugs that target lipid 2. So what is this, the target lipid 2? As all of you know, bacteria are surrounded by peptidoglycan, which is essential for their survival, their diary diet. And peptidoglycan is composed of peptide sugars here in blue that are connected by short peptide bridges in yellow. And peptidoglycan is a constant turnover, and it is maintained by its precursor molecule lipid 2 that you see here. And lipid 2 carries one sugar peptide head group that is connected to a conserved pyrophosphate and a complex, very long lipid type. And lipid 2 is assembled here in the cytoplasm, then flipped across the plasma membrane, where the head group is chopped off and integrated into peptidoglycan, and the rest is cycled back for the next round. And this lipid 2 cycle is essential. Any drug that interferes with it is an antibiotic. And this tenet was famously validated by vancomycin. And vancomycin used to be an antibiotic of last resort, but you're chemists, biochemists, structural biologists, you know that peptides can be readily modified, and that's why many bacterial strains are resistant to vancomycin these days. To this end, we work on smarter drugs that target here the pyrophosphates. And phosphates cannot be mutated, and that's why this binding mode is very refractory to the development of antimicrobial resistance. And it's also very powerful. Over the last decade, a number of excellent antibiotics have been discovered that target lipid 2 or similar molecules. And the most famous of these molecules is presumably Teixobactam. And Teixobactam was discovered by Kim Lewis from Northeastern University, Boston, and the company Novobiotic Pharmaceuticals, also from Boston. And these people are able to isolate natural products from so-called previously uncultured bacteria. And so they isolated Teixobactam from a previously uncultured bacterium, and this discovery made a big fuss and was heralded as a wonder drug, and that's of course completely exaggerated, but Teixobactam has a real perspective, and clinical trials will start soon. So what is Teixobactam exactly? It's an 11-residue peptide that features many non-canonical elements, including a couple of D amino acids and the cyclic so-called DEPC cycle here at the C-terminus. In this DEPC cycle, we have a very rare cationic amino acid called enduracylidine that has only been encountered 3 times in nature. And in this original Nature paper, it was also shown that Teixobactam targets lipid 2. So what else was known? It was known that if you chop off the linear N-terminus, then you lose the activity. And furthermore, it was also shown that you cannot tamper with the order of D or L or polar or apolar amino acids. All this completely kills off the activity. So a lot was known before we started, and you wanted to contribute. So what I didn't tell you so far is our core expertise is solid state NMR. And in order to do solid state NMR, we need isotope labels. And that's really tough for such a complex peptide, and you also cannot produce it recombinantly because it's non-ribosomal and it comes from funky bacteria. The cost of synthesis for such a complex peptide to isotope label is very hard. And that's why we initially teamed up with Ishwa Singh from Liverpool University, and Ishwa had shown that this funky residue here can be replaced by conventional hydrophobic aminos. This keeps a good deal of the activity, and it slashes the cost of synthesis. So initially we worked on these synthetic tioxobactam analogs, and in order to make them amenable to solid state NMR, we introduced 6 isotope labeled residues here as these residues of the L-amino acids. And this here is already the bound state, so tioxobactam bound to lipid-2 in membranes. And we count 1, 2, 3, 4, 5, 6 signals corresponding to these 1, 2, 3, 4, 5, 6 residues. And this is really the bound state that was completely different in the free state. And we can readily confirm that this is the bound state using phosphorus solid state NMR membranes. So this here is the free state, the phosphorus solid state NMR spectrum and membranes. We count 3 signals. The 2 smaller signals here relate to the pyrophosphate, and the big signal relates to the bulk lipid. And once we add the drugs, we see a major shift in the pyrophosphate, confirming complex formation. So let's look into this complex at high resolution. And we first wondered about the secondary structure of tioxobactam in the complex, and we can readily get the secondary structure from this beautifully resolved 2D carbon-carbon spectrum. This works as follows, which simply makes a difference between the random coiled textbook chemical shift and the experimental chemical shift. And if this difference is positive, it means alpha helix, and if it's negative, it means beta strand. And you readily see that the N2 is the beta strand conformation in the complex. And then we wondered about the supramolecular arrangement of tioxobactam in the complex. Again, we acquired 2D carbon-carbon spectra with different magnetization transfer times. With a short transfer time, we only observe intra-residual contacts within the same residue. And with a long magnetization transfer time, we observe inter-residual contacts between different residues. And indeed, you see many more contacts here with a long transfer time. And I'm going now to superimpose signals in these green boxes, and please focus on signals with red labels. These are the interesting long-range contacts across many residues. And we see a long-range contact between residues 2 and 6, 3 and 7, and even 2 and 7. And if you consider that the textbook C alpha to C alpha plus 2 distance in the beta strand is around 6 angstrom, then it would mean that the distance between residues 2 and 7 is 15 angstrom. And ask any of the many NMR spectroscopists of your choice, this person will tell you that's not possible with NMR, and you can only explain these distances if they're intermolecular between different tioxobactam molecules, and therefore you need these antiparallel beta strands. And we could readily confirm that tioxobactam oligomerizes up on target binding using fluorescence spectroscopy and pyrene-tagged lipid-2 molecules. This is a bit like Fred. Once 2 pyrene molecules come close to each other, they emit a unique X-mer signal at 490 nanometer. And this is exactly what we observe once we added tioxobactam to pyrene-tagged lipid-2 in membranes. We see the dose-dependent buildup of this X-mer signal. So tioxobactam oligomerizes up on target binding. Let's look into these oligomers at high resolution. And we first wondered if the depth cycle of tioxobactam really directly coordinates a pyrophosphate. This was conjectured in the literature, but there was no data available. So to get this data, we acquired this beautiful result, 2D phosphorus proton spectrum. And again, the phosphorus dimension is simple. We have all these 3 signals, the big one from the bulk lipids, and the 2 smaller ones from the pyrophosphate. And only the pyrophosphate talks to the amino protons of tioxobactam. Next, we needed to annotate these signals. And therefore, we needed to realize that this proton dimension and that proton dimension are exactly the same. And then we can simply plot the assignments on the spectrum, and then we see that these signals match very well with residues 9 and 10 of the Dempsey cycles, and the Dempsey cycle really does directly coordinate pyrophosphate. And I also want to stress that this spectrum was actually acquired in Florence and supported by both INEX and Instruct. So thanks very much for this. So, what we also critically need to understand is the role of the sugars and the pentapeptide. And to remember from vancomycin, especially the pentapeptide is important. It's here the bacteria introduce lots of modifications that can cause antimicrobial resistance. So, in order to make this happen, we needed to isotope label lipid-2, and this was done by a former PhD student, Joe Silver, and my collaborator again, Brooking. This worked very well. You see here the pentapeptide, and these are the sugars. And with this you can now acquire 2D carbon-carbon spectra of the complex in which both tioxobactam and lipid-2 are carbon-13 labeled and derive unambiguous interfacial contacts. So here are all the interfacial contacts that we could derive plotted on the primary structure of tioxobactam. And first, you see that all the interfacial contacts cluster on the Dempsey cycle. So the Dempsey cycle is really the binding platform. And second, we see that almost all contacts relate to the Murmann sugar. And the Murmann sugar is a sugar that is directly attached to pyrophosphate. So Murmann must be directly at the interface. For the second sugar, we barely observed any contact. So the second sugar must be distal from the interface. And then curiously, in this spectrum, we did not observe any signals for the pentapeptide. And now we need to know that this is a so-called dipolar spectrum. In a dipolar solid state NMR spectrum, you can only observe rigid signals. So if we don't observe the pentapeptide, it's most likely highly mobile. And indeed, we could confirm this with a so-called scalar spectrum. In a scalar spectrum, you can only observe highly mobile signals, and here we only observe the pentapeptide. So the pentapeptide is highly mobile at the complex. What have we learned as well? We have learned that the Dempsey cycle directly coordinates pyrophosphate, Murmann is at the interface, Glückmann is distal from the interface, and the pentapeptide is highly mobile and plays no role for the interface. And that the pentapeptide plays no role for the interface is the very reason why tioxobactam is so refractory to the development of antimicrobial resistance, because it doesn't care for all the modifications that bacteria introduce here. So this is a structure model of the complex in yellow, the beta sheet of tioxobactam, and we see that the Dempsey cycle tightly coordinates pyrophosphate. And what I enjoy most about this complex is that it explains the stereochemistry and the membrane topology of tioxobactam. In the complex, there's a clear-cut separation of lipophilic and hydrophilic residues. Lipophilic ones point to the membrane and hydrophilic ones to the water. And this separation is only possible with the stereochemistry. If you imagine a hypothetical all-else stereochemistry, you ended up with some lipophilic residues that face the water and some hydrophilic residues that face the membrane. So you need this stereochemistry to get this topology. So this was what we published a while ago, and in this manuscript we made one more interesting observation. So we knew that tioxobactam oligomerize on target binding, and we wondered, hey, can we even observe these oligomers at the macroscopic level? And therefore we used a tagged or fluorophore tagged lipid-2, and you see here the uniform distribution of this tagged lipid-2 on the surface of a liposome. And once we added tioxobactam, we observed these massive clusters. And this was really striking, but we had no idea what this meant. And in the end, there were two major open questions. First, what is the role of these clusters? And second, what is the role of the non-canonical residues, especially what is the role of endurosylidine, the funky residue that has only been encountered 3 times in nature? And then we were simply very lucky. So on the same day that we published our manuscript, so I had a very pleasant phone chat with Kim Lewis, and Kim, together with the company Novabiotic, is a developer and discoverer of natural tioxobactam. And soon after this pleasant phone chat, we got a few milligrams of uniformly carbon and nitrogen labeled natural tioxobactam. And I hope you appreciate from this data that natural tioxobactam in membranes in complex with lipid-2 gives outstanding data that we can readily annotate each and every signal, including signals for the non-canonical residues. So here this is endurosylidine, the funky residue, and this is D-and-phenylalanine, D-and-metyl-phenylalanine at the end-terminus, which is also very important, as we will see later on. So long story short, natural tioxobactam also forms beta sheets upon complex formation, and they can also determine the interface using carbon-13 labeled lipid-2. Here we have zoomed into the endurosylidine side chain. In blue is carbon-13 labeled lipid-2, and in red with unlabeled lipid-2. And all the blue contacts with no red correspondence are unambiguous interfacial contacts between the Murmann sugar of lipid-2 and the endurosylidine side chain. This here is a structure of the complex inside the beta sheet of tioxobactam, and we see that the Dempsey cycle tightly coordinates the anionic pyrophosphate group, and we see that endurosylidine forms out specific hydrogen bonds with the Murmann sugar. And interestingly, we also observe here that D-and-methylphenylalanine was the N-terminus of an adjacent tioxobactam molecule forms out contacts with lipid-2. And this means that in the complex, each lipid-2 molecule is coordinated by 2 tioxobactam molecules, and this is one of the reasons why tioxobactam needs to oligomerize to get this excellent killing activity. And thereby, these later explain the role of the non-canonical residues in natural teixobacter. But what about the other major open question? What about these clusters? Natural teixobacter also oligomerizes and forms these clusters in membranes. You see this very nicely in these bright field images. And this time, we even observe membrane perturbations at the site of the clusters. But this all happens in liposomes, and that's nice to know, but at the end we need to understand the mechanism in bacteria. And therefore we teamed up with James Norwick from UC Irvine and also Harry McGilvery from Utrecht to help us with super resolution microscopy. And here James provided us with a fluorophore type teixobacter that we could use to study cluster formation in bacteria. And indeed, briefly, shortly after the addition of teixobacter to bacteria, we observed these massive clusters which unambiguously confirms that cluster formation is an intrinsic part of teixobacter's killing mechanism. So far, we have looked at these clusters at submicrometer resolution and at atomic resolution. But to truly understand the nature of these clusters, we needed to close this resolution void, and we did this with atomic force microscopy in collaboration with Walter Roos from Groningen University and this fantastic postdoc Sir of my team. And I'm really very fond of these data. I think they are self-explanatory. What you observe here is that teixobacter forms fibrils upon lipid-2 binding. Fibril formation matches very nicely with the solid state and mass structure. And you see these fibrils emerging after about 10 minutes. And also the time scale of fibril formation matches very well with the emergence of clusters in bacteria. So teixobacter forms fibrils upon lipid-2 binding. And the last thing that we needed to clarify was this here, the membrane perturbations at the site of the clusters. And here we exploited that atomic force microscopy is a gorgeous method to determine the heights of the membrane. You can do this at almost atomistic resolution. So we're going now to look at the heights of the membrane here at the site of the cluster, here in dark orange, and here in dark blue. And you will see that this is supposed to be a video, doesn't work. You're going to see that again on the next slide. So what you would see is that here at the site of the membrane is heavily thinned by 1 nanometer. You see this also here. This is an empty simulation. It looks exactly like the AFM later. So teixobacter thins the membrane by about 1 nanometer, and we tried to understand this with MD simulation performed by my PhD student Francesca. And she took the solid state and a mass structure and simply simulated this for a few hundred nanoseconds. And indeed, after a few hundred nanoseconds, we could reproduce this membrane thinning in very nice agreement with AFM data. So how can we rationally explain this membrane thinning? And there are most likely 2 reasons for it. First, at the site of the clusters, the hydrophobic underbelly of teixobacter pushes the polar lipid head groups away. And more importantly, at the site of the clusters, we have a very high concentration of lipid-2. And lipid-2 has this funny, strongly non-lamellar lipid tail that's 55 carbons. You see that here, the lipid-2 tails deeply reach into the other half of the membrane, and this compromises membrane stability. And it compromises the membrane stability so much that you actually lose the membrane potential. So this means that teixobacter actually uses 2 killing mechanisms simultaneously, and we call this a 2-pronged attack on the cell level. So teixobacter not only blocks peptidoglycan synthesis, but it also compromises membrane stability. And the great thing is that teixobacter only compromises the stability of membranes that contain lipid-2, and only bacterial membranes contain lipid-2. Thereby, we also elegantly avoid this toxicity issue. All right, now I'd like to tell you the story of clovibacter. Clovibacter is a brand new antibiotic isolated from previously uncultured bacteria. Again, this was discovered by Nova Biotech and Kim, and together with Tanja Schneider from Bonn in Germany, we looked at the killing mechanism. And clovibacter is a potent antibiotic with activity against a broad spectrum of superbugs, including in animal models of infection. And clovibacter also targets lipid-2. And it targets lipid-2 using a new binding motif that we call hydrophobic cage. We previously called this a hydrophobic glove, so that's why we still have this illustration, but meanwhile we call it a hydrophobic cage, and that's where the name comes from. Clovi means cage in Greek. And I will show you how this hydrophobic cage makes clovibacter extraordinarily robust against the development of antimicrobial resistance. Clovibacter also uses a supramolecular mechanism. You see here, or you will see here, a clovibacter fibril emerging. And curiously, for all of the 2 binders that we studied, we observed these supramolecular mechanisms. And meanwhile, we try to generalize this and we call these supramolar mechanisms the Welcher mechanisms. So why do lipid 2 binders use the Welcher mechanism? Well, if you have one drug that binds a target, you end up with a weak temporary interaction. But if many drugs come together and traps the target in the superstructure, the target can't go anywhere, KO drops dramatically, and you end up with a binding that is ultra tight and virtually irreversible, and that's why these drugs use the Welcher mechanism. Alright, so this here is the chemical structure of clobibactin, this is the assembly domain, and this is the binding domain. The binding domain is the Dempsey cycle that coordinates pyrophosphate of lipid 2. And if you look carefully, then you see that all residues of the binding domain are hydrophobic. And this is really counterintuitive because hydrophobic side chains cannot specifically interact with pyrophosphate, and this is key to understand the binding. We use solid state NMR to study the dynamics of clobibactin and the complex. And we did this for each and every residue here. The higher the bar, the higher the dynamics. And first, you see that the dynamics of bound clobibactin are much higher than the dynamics of bound tixobactin. And second, you see strongly elevated dynamics here at the hinge of the binding motif. And this means that the binding motif is very flexible, and this flexibility, this plasticity, enables the binding motif to snugly wrap around pyrophosphate of several indispensable cell wall precursor molecules that all have pyrophosphate in common. And this multi-target attack mechanism is the very reason why clobibactin is so robust against the development of antimicrobial resistance, because the more targets you have, the less resistance can occur. This is a little summary, so I've shown you that vancomycin is prone to resistance development because it targets a pentapeptide which can be easily modified. On the other hand, tixobactin is very robust to resistance development because it targets a conserved pyrophosphate sugar motif. And finally, clobibactin is most robust against resistance development because it exclusively targets the immutable pyrophosphate group. And together with the high potency of clobibactin and its low toxicity, we believe that clobibactin could be an interesting template for the design of next generation antibiotics. And I also want to stress that all the NMR data for this project were required on our Bruker 1.2 GHz magnet. You see that the resolution on the 1.2 magnet is much, much better than on a 700 magnet, and this increase in spectral quality was really instrumental for the success of this project. And also here, phosphorus NMR data were required, and fluorine supported by Instruct. Alright, so for the last few minutes, I will speak, surprise, on another antibiotic. And this antibiotic is called Plexaden. Plexaden is not brand new at all. It was discovered 20 years ago by the Danish company Novozymes. And the original paper has been cited 800 times and is really a hallmark of the antibiotics field. So myself, I've been working on Plexaden for 8 years. This is basically the first project that I started once I became independent. And today I'm really super excited because just this morning, after 8 years, we have finally published our findings on Plexaden, so let me show you what we found. So this is Plexaden. It's a little 40 residue peptide. It's so-called defensin, a host defense peptide, which is part of the innate immune response. You find this in many higher organisms, and it's conserved, and the fold is indeed conserved in many higher organisms. Plexaden itself comes from fungi. Then, in 2010, the Danish company showed that Plexaden targets lipid 2. There's even an NMR structure model in this paper, and in this structure model, they show that lipid 2 binds here somewhere close to the beta sheet. And this is really an amazing paper. I'm really fond of this paper, and with the data that they had back then, I would have come to exactly the same conclusions. Nevertheless, meanwhile, we know that there are certain functional observations that do not align well with this structure model. And the most prominent observation is that if you change residues in this anionic patch, the function of Plexaden changes, and there is no explanation for this available. Well, if you have an anionic patch, then you automatically think about cation binding, right? And we conceived a very simple approach to probe cation binding. We simply acquired a solution in the mass spectrum of Plexaden in the presence of manganese cations. And manganese is a paramagnetic ion, so any residue close to manganese will disappear from the spectrum. And indeed, there are lots of residues that disappear from the spectrum, and if we plot the residues that disappear onto the structure, then all these residues are here around this anionic patch. And the anionic patch is a bivalent cation binding site that binds specifically to bivalent cations and especially calcium. And if you then measure the binding affinity between plexidin and its target LIPA2 in the presence of calcium, then you see a massive increase in affinity by more than a factor of 100, and calcium also massively increases the activity of plexidin. What happens here is that calcium binds to the anionic patch, and this allosterically changes the conformation of this loop, and it's actually here where LIPA2 binds. But meanwhile, we know that LIPA2 binders use the Velcro mechanism, right? And guess what? Plexidin is no exception. In a few seconds, you will here see a supramolecular carpet of plexidin emerging. Here we go. And the supramolecular carpet sucks up all the LIPA2 from the membrane. And this supramolecular carpet mechanism is highly calcium dependent. In the absence of calcium, you have this jagged surface. The surface is jagged because the superstructure is highly disordered. And in the presence of calcium, you have this smooth surface. And this means that the superstructure is perfectly ordered, and you need this perfectly ordered superstructure in order to kill bacteria efficiently. So this just came out this morning. It was really a hell of a project, 8 years of work, and a really big congratulations to the two lead PhD students, Sherazada and Mike. I think we're all happy that this is over. And this here is a project from the press release where the 4 first stunts on top of the magnet in our lab course. I think that's the first time that I've won a lab coat in 10 years, but yeah. Anyway, so this is my last slide. And I told you that bacteria use the Velcro mechanism because it makes the binding irreversible. And I said this, but I didn't show you any proof because I didn't have any proof. Now we tried to get this proof. And what we did in order to get this proof was the following. So we incubated bacteria with a fluorophore type surfactant. And then we waited for 45 minutes and imaged. And then we observed these clusters here. So far so good, we have shown this already, right? We've shown that before. And what we did afterwards is we so roughly washed these bacteria in buffer. And we did this many times in order to get rid of all the free types of bactin. So if we get rid of all the free types of bactin, we heavily favor the destruction of this complex. We heavily push the equilibrium towards destruction of this complex, unless the binding is irreversible, right? And we did this many times, we washed for 5, 6 times, and then we imaged the complexes again after 4 and 18 hours, and after 4 and 18 hours, these complexes are still intact. So that means that these superstructures really form irreversibly on relevant biological timescales. So this is maybe my only take-home message, the Velcro mechanism, and I don't think that this is something isolated for antibiotics. You see something similar for antifungal drugs such as amphotericin, and most likely this is much more widespread in antibiotics and antifungal drugs. With this I'd like to come to the acknowledgements, so I've thanked already all my amazing collaborators, and I would like to thank the groups, they're all superheroes, and I'd like to thank Instruct again for the organization of the meeting, and thank you for your attention. Thank you.